This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 113, recorded December 24th, 2010. Hi, everybody. I'm Vincent Racaniello, and it's time for TWiV. Joining me today from Western Massachusetts is Alan Dove.
Good to be here.
How you doing, Alan?
Doing okay. How about you?
Cold as usual?
Oh, yeah, it's chilly, and we got a few snow flurries yesterday, which was a nice prelude to Christmas.
I thought you were heading down to the city soon.
Yeah, we'll be going down
Later?
to the suburbs of the city anyway, but we'll do that later this afternoon.
Oh, very good. Our next host from North Central Florida is Rich Condit.
Hi, fellas. How's everybody?
75 degrees?
No, it's chilly here. Chilly for us. 61.
Here it's really cold.
Right.
But at least it's
61
sunny.
Here, it must be really cold there.
Yes,
Yeah, it's sunny.
I have to tell everybody, yesterday Alan and I were trying to, Alan Dove, were trying to sort out the Skype issues. Skype has been down this week. So Alan said, let's do a check. And Rich said, I'm sorry, I just teed off. Nice. Our special guest today is the head of the retrovirus assembly program at the National Cancer Institute, Alan Rein. Welcome to TWiV.
Hello, Vincent. Hello, other hosts. It's very nice to be here.
Thanks for joining us. We appreciate it. We thought we would have you on to clear up all the questions about XMRV.
It will be a pleasure.
I'm glad of that.
And we can all go home happy.
Yep. On Monday, a clutch of papers were released in the journal Retrovirology, and I said, we have to get somebody to talk about these with us. And Alan's name came to mind because you had just published a paper not too long ago, which you sent to me, and that's why one of the reasons why you're here. But we haven't talked about XMRV since, I think, the low alter paper, and we have talked about the virus and its potential involvement in chronic fatigue syndrome and prostate cancer quite a bit here on TWiV, but the low alter was the last we discussed. So I wanted to start, well, I guess we should first start with telling everyone that you have a lot of experience in studying retroviruses, right?
Yes.
I'm looking at your page here. You have been, I guess you've been at the NCI since 1976, right?
More or less.
So you have worked on retroviruses that entire time?
Oh, yes.
And which ones? All of them?
Well, for most of that time, murine leukemia viruses. Of course, there was no HIV in 1976.
Right.
And then I've also done a lot of work on assembly of HIV.
All right. So you have a lot of experience with the murine
Yes.
leukemia viruses, which is, of course, the same kind of virus that XMRV is, right?
Mm-hmm.
Okay. So and you're head of the assembly section, so you've got a lot of experience. You've published a lot of papers, so you're an expert, and you know infinitely more than we do about this virus. I just want to point that out to everyone.
Yes.
So I'm just going to sit back
Okay,
and
so
listen to
that's
this.
settled.
You're going to sit back and listen, Rich?
Yeah.
You know Alan a bit, don't you, Rich?
Well, we met for the first time this last summer, coming back from a FASEB meeting on virus assembly. Good story. The bus broke down. So we had to get out, and we were standing by the side of the road. Somebody noticed some poison ivy.
I noticed the poison ivy and
Was it
pointed
you?
it out to this German investigator who was not familiar with poison ivy. I
Right?
warned her
No, there
about
was a
it.
discussion of poison ivy, and we got back on this rickety old bus that rescued us to take us to the airport, and we start singing the song, Poison Ivy. And in fact, I was actually going to use that as a pick of the week because it turns out that it has bugs in it. Measles makes you bumpy, mumps will make you lumpy, chicken pox will make you jump and twitch. Common cold will fool you, whooping cough will cool you, but poison ivy
Poison
won't
ivy
make
sure will make you itch.
Who did that
It
song
turns
originally?
It turns out that Alan knew all the lyrics to this song. I couldn't believe it. Not only that, but we had several other songs going on. He knew all those. I told my daughter about this. I said, he knows all these 50s songs. She says, of course. He's a retrovirologist.
Oh.
great.
Oh, hey, that's my job.
She got me
I
nailed.
know, Alan, but I couldn't help it. I thought that was just too good.
That is good. Who did Poison Ivy originally?
The Coasters sang. Songwriters,
Coasters,
Lieber,
right?
Coasters, yeah.
Joey Lieber and Mike Stolper.
Let's start with the low alter paper. We talked about it, but I was wondering, what were your thoughts? They found a different virus from XMRV in specimens from patients with chronic fatigue syndrome.
Well, my thoughts are, they didn't find a virus. They found sequences. And the sequences are sequences which are present in normal mouse DNA, uninfected mouse DNA. And in fact, they are sequences of endogenous murine leukemia virus genomes, which in general are defective. They have been there for some unknown length of time, and they don't produce virus. And they managed to amplify these sequences using their PCR primers out of their DNA preparations.
Now, they went through a lot of trouble to try and show that there were not mouse nucleic acids contaminating their specimens, right?
Yes, they tested for mouse mitochondrial DNA, and so that's very important. I think the level of mitochondrial DNA varies from one cell to another, so it may not be the best way of assessing the level of mouse DNA contamination in a DNA prep. And two of the new papers that just came out used a different assay for mouse DNA, which is for intracisternal A particle sequences.
Right.
This assay is specifically for nuclear genomic DNA of the mouse, and I believe is quite a bit more sensitive than the mitochondrial DNA assay.
And that probe is actually a piece of a retrovirus in itself, isn't it? It's a highly repeated sequence.
It's a highly repeated sequence, and I guess it's a retrotransposon.
Right.
Mm-hmm.
So it probably would be a good idea for Alter to go back and reprobe his samples with the IAP probe.
I think that would be a very good experiment.
You have no idea whether he's doing that or not, right?
No, I don't know.
Because you're at the NCI, which is separate from the rest of the NIH, right?
It's a big place, yes.
They also didn't get complete genomes from these patients, which now I know in your paper you mentioned that the sequences he published were identical to mouse sequences. Is that right? They're absolutely 100% identical?
I believe so.
And that you wouldn't expect that, especially there was one case where I think he had samples from the same patient 15 years apart, right?
Yeah.
So this has been a question which has been an issue since the beginning, and that is how much variation would you expect? And I don't really know, because with HIV we're kind of spoiled that you get huge amounts. But what could be the minimum amount?
I don't think we know. I mean, you're right. HIV, which of course is replicating in astronomical levels every day in an infected person, gives tremendous amounts of variation. But murine leukemia virus has a somewhat different biology, and I don't think we know how much replication there is in a viremic mouse, let alone a human. So I don't think the experiment's been done.
So the fact that there is so far observed small amounts of variation, that it by itself does not rule out the replication of these viruses in humans, for example.
Yes, in my mind, to my mind, it does not rule it out. And it's very important that you can't. Facts about one retrovirus don't necessarily apply to another in this case.
Right.
The biology is different from that of HIV and not as well characterized.
Not too long ago, you published a paper where you looked for the presence of XMRV in prostate tumors. Mm-hmm. Can you tell us about that?
Sure. This was a collaboration with the lab of Angelo DiMarzo at Johns Hopkins, who is in the urology and pathology departments. And we did PCR looking for XMRV sequences in prostate cancer DNAs. And, well, very briefly, we did not find any. And we had a couple of ways of validating our negative results. First of all, we quantitated our ability to detect XMRV in 22RV1 DNA. This is a human cell line that is producing XMRV. And second of all, it was duplex PCR, that is, every PCR well was simultaneously amplifying a cellular gene, CCR5, and quantitating that. And so we know that the amplification was going fine in every single well. And nevertheless, in all our prostate cancer samples, the XMRV assays were completely negative.
And this wasn't just a couple of samples either, right? This was 800.
So the PCR was done on, I think, between 1 and 200. And then the larger number was assayed by immunohistochemistry. And this was done in Angelo's lab with our Anasera. So in this case, we had Anasera that had been made years earlier against two distinct proteins of Maloney murine leukemia virus, one an internal viral protein, capsid protein, and one an external viral protein, GP70. And the immunogens used to make these rabbit polyclonal Anasera were proteins that had been purified by HPLC. And so Angelo used these Anasera to stain formalin-fixed paraffin-embedded sections of prostate cancer, and his assays were about 800 samples. And again, to make a long story short, all the prostate cancer assays in his hands were completely negative, and the validity of the assay was demonstrated by staining either cells that we had transfected with an XMRV plasmid from Robert Silverman, or infected cells such as 22RV1. So in every assay, both sera stained either the transfected cells and the infected cells, but never stained any of the prostate cancer samples.
Now, you specified that the antigens used to make those antibodies were HPLC purified. That's important, right?
I think it's very important. I think so. These retrovirology papers are about how you can get false positive PCR results from, contamination with minuscule amounts of mouse DNA. But another way to get false positives is if your anaserum reacts with cellular proteins. And so I think a possible problem in the positive immunohistochemistry results that have been reported is the following. Dr. Singh's lab, as I understand it, made XMRV in human cells, took that XMRV and put it into rabbits. That's how they made their anti-XMRV Anasera. And then they took that Anaserum, and lo and behold, it stained a large fraction of prostate cancer sections. But it is known with both HIV and murine leukemia virus that retroviruses actually incorporate cellular proteins into the virus particle itself, so that even a perfect purification of the virus would not get rid of these cellular proteins. So I think it's very possible that Anaserum has antibodies not only against viral proteins, but against cellular proteins. That is, human proteins. And so when it is put on human cells, it stains them, but that does not necessarily mean it is staining viral proteins. phenomenon of incorporating cellular proteins into retrovirus particles has quite a history. It really confounded a lot of the early vaccine trials, monkey trials against simian immunodeficiency virus.
I think you had mentioned in your paper you obtained a few of the positive samples from Ela Singh, the samples that were positive by IHC, and in your hands they were no longer positive.
That's right.
Using your antisera, right?
That's right. Mm-hmm.
I think that's an important point, that the antisera have to be good quality.
Mm-hmm.
And since your paper came out, there was another paper from Baylor, published in the Journal of Infectious Diseases, in which they did detect XMRV in normal and tumor tissue from patients from the southern U.S.
Mm-hmm.
Have you looked at that?
I have not read that paper closely, and I think the key question is what were their controls to rule out contamination, but I really can't comment on the paper.
They have they looked in normal and tumor tissue from some of the patients, and a certain fraction they could find XMRV in both normal and tumor tissue, which they say suggests that infection may precede cancer onset. Controls, what sorts of control? You mean a water control for mouse contamination with mouse nucleic acids?
Well, that's a good control. That's a place to start. Or bacterial DNA. I mean, just some DNA.
Let's see. So what they did was look for proviral DNA.
Real-time PCR. Nested PCR assay.
Nested PCR. So, Alan, your assay was for viral RNA, right?
No, it was DNA.
So it's proviral DNA.
Mm-hmm.
And that's what they did here. They looked for proviral DNA. And let's see if I can find a control.
The master mixes for each PCR reaction is tested for sensitivity and nucleic acid contamination by incorporation of positive and negative controls. Positive control, 3 separate reactions, 100 micrograms of XMRV-infected LNCAP cells. Negative controls, 3 separate reactions in water in place of DNA template. This is the paper. Is this the paper where they point out that they could see it if they use 600 nanograms of DNA, but not 100?
Yes,
well?
I think that's this paper.
Right. So whether or not they got positive results was dependent on how much DNA they put in the PCR reaction as well. But it looks like their major control is just a no DNA control.
Mm-hmm.
So is that adequate?
Well, how about some negative human DNA, some HeLa cell DNA? I'm sorry, I have not studied the paper, so I really shouldn't comment on it.
So we'll put a link to it. It's a as I said, study of southern U.S. patients, 144 patients. Now, where were your patients from? Baltimore area?
Mostly, yes. Mm-hmm. Although there was others as well. Like we got some from Metastasis, which were from the University of Michigan.
Mm-hmm.
But mostly from around Baltimore, yes.
So do you think there could be geographic restrictions to this virus in prostate cancer?
That's possible. I guess I don't think it's very likely, but
because I don't know. We're just trying to struggle with how to reconcile your results with findings like these, and there are certainly other negative results. There are other positive results as well.
Well, it sounds to me like based on these papers that we're going to feature today in retrovirology, that an important control to put in all these kind of studies is some sort of assay for other mouse DNA that could potentially be contaminating the samples.
Yeah, I think this intracisternal aparticle assay is a real step forward as an extremely sensitive assay for mouse DNA contamination.
Right,
now,
and in at least one of these, maybe 2 of them, they also found that they had samples that tested positive for mouse DNA contamination by IAP, but tested negative by mitochondrial DNA, which was the previous standard.
Yes. Mm-hmm.
So the previous study should go back to their their samples and look with
Yes.
this assay.
And Alan, there seems to be a lot of there's a lot of variation in exactly the PCR assay used, whether it's real-time PCR or just looking for fragments, and whether or not stuff is sequenced, whether it's nested PCR or straight PCR,
Sure.
what primers are used. Can you? It's a lot of stuff, but can you generalize as to what might be better or worse, or what differences makes?
That's a good question. Real time obviously has the advantage of quantitation, but nested PCR is certainly extremely sensitive. A point I did want to make, which I didn't quite get around to, is PCR, of course, can be, you can miss what you're looking for because of sequence polymorphism. That is, you make primers against, let's say, a published XMRV sequence, but maybe the XMRV in your sample is not identical to the published sequence, and maybe for that reason you'll fail to amplify it. And I think that is, a risk in the PCR and a source of possible false negatives, but that objection does not apply to the immunohistochemistry because the antibodies that we used were broadly reactive. They were made against one murine leukemia virus, but we showed they reacted against XMRV. And so I don't think that they are sensitive to, minor sequence variations, polymorphisms in the virus. And so although that assay, is not quantitative like PCR and maybe not quite as sensitive, on the other hand, it is going to find XMRV regardless of polymorphisms.
So as in many experiments, using very different technical approaches to arrive at the same answer is important.
Yes, absolutely. Yeah.
Do you have any other plans to do additional work on XMRV?
Well, it seems to me that where the field is some people find it and some people don't, and basically every lab has their own assay. And I think we cannot move forward as a field. We cannot resolve the contradictions until there are assays which can give consensus. Different labs can test the same sample and say, this one is positive, this one is negative, this one has a lot, this one has a little. And so there is an attempt underway to essentially to distribute samples to different labs, and I'm hoping to take part at NIH, and I expect to be part of this effort. It seems to me that's the next step before the contradictions can be resolved.
Now, in sending the samples around, given what we see in these 4 retrovirology papers, one of the things that stands out to me is how minuscule an amount of contamination can give these positive results, or apparently positive results. So as the samples, I can imagine if you receive these samples in your lab, you presumably you're going to have some kind of containment for them so that you don't all of a sudden start getting all of your tests coming out positive, right?
Well, yes, but at least if they're sent with no virus in them, then if you find virus when there shouldn't be any virus, at least you have a problem.
Right.
Yeah, it seems to me a study like that ought to be done blinded as well, or at least part of it.
Oh, absolutely. That is crucial. Yes, that is crucial, that it must be done blinded. John Coffin did a wonderful Gedanken experiment. He did the calculation. If you took a drop, 50 microliters of mouse blood, you put it into his swimming pool, 30,000 gallons, mixed well, and took out a mil from that swimming pool, you would be able to find murine leukemia virus sequences in that mill. That is the level, of murine leukemia virus sequence in mouse biological materials.
And apparently, one of these papers, they had samples that had been collected by the same lab, probably by the same people, just at different times. and that alone was enough to skew the results, where some were contaminated and some weren't.
Mm-hmm. And of course, another of those papers showed that the hot start Taq polymerase from Invitrogen was contaminated with tiny amounts of mouse RNA, which gave positive results in XMRV PCRs, RT-PCRs.
Yeah,
Right.
so for the listeners, we haven't actually run through these. We've been talking about 4 papers, but we haven't said what they are. Why don't we just quickly summarize what's in each of these 4?
All right.
Good idea.
So, Alan, you said one showed that there's a... I meant Alan Rein, actually.
Oh, sorry.
Alan Rein just said there was one paper that showed that a PCR kit was contaminated with mouse nucleic acids, right? So that was a Japanese study where their water controls were coming up positive for XMRV, right?
Right.
So they traced it to, I think, a monoclonal antibody in one of the kits, which is against the DNA polymerase, which...
Right. It's a mouse monoclonal antibody.
Right.
That's right.
So you purify from a mouse cell and it still has nucleic acids in it.
That's right.
Now, of course, that doesn't mean that all the previous studies are contaminated in this way, but it just says you have to be really careful, right?
Right.
Right. So that's one of them. And then there were 2 on which John Coffin was an author, basically dealing with contamination of samples with mouse DNA.
Yes.
I think one was a prostate cancer study and the other was a chronic fatigue syndrome study.
I think so. I'm looking for the papers. But anyway, yes. And those 2 papers both used the intracisternal aparticle assay to detect mouse DNA.
Yeah, and their point was the IAP assay, as you said, is much more sensitive.
Mm-hmm.
I guess that's because there are more copies of IAP.
That's right.
In the genome, right?
Yes.
Now, what was interesting in those studies is that they got positives which were in the ballpark with some of the other studies at a level or at a frequency, but they were all accountable by the presence of contaminating mouse DNA.
That's right.
Where did it come from? Do we know?
No, I don't think we know.
I'll wager a guess and say it came from mice.
Good, Alan. Thanks.
But
That's very helpful.
So,
that was the other Alan.
Yeah, I mean, how do mouse sequences get into? So I guess it could get into reagents. It could even be in water. Let's consider this. Here in New York City, our water comes from reservoirs upstate, right? And I'll wager that those reservoirs have mouse feces and urine in them.
The Food and Drug Administration mandates what levels of rodent droppings and insect parts are permissible in our food and water.
Mm-hmm.
And the levels are not
and they're not zero.
So then the water that I use in my lab, we put through a reverse osmosis system, and the final step is a 0.2 micron filter, which, as far as I know, will let most viruses through except Mimi viruses, right?
Yes, we're talking about nucleic acid, I think, not virus.
Well, it could so the nucleic acid could go through. So it could be that even your water control could have mouse nucleic acids in it, right? So you really should test for this IAP.
Also, if anybody in your lab or in your building does mouse work, I can imagine, somebody goes up to the mouse lab, comes back down to their bench and starts pipetting.
Sure.
If it only takes a cell.
Yeah.
Takes much less than a cell.
Right. You just need a fragment of nucleic acid.
So the calculation I did was a millionth of a microliter of mouse blood will have detectable murine leukemia virus sequence in it.
And it's not just blood. Dander presumably would have some detectable sequences too.
Mm-hmm.
So one of these papers in addressing this question of where did it come from simply kind of throw up their hands and say, I don't know.
Mm-hmm.
And the other paper says that they're talking about assaying blood happened either during collection of blood, isolation of peripheral blood mononuclear cells, or during a preparation of DNA from the cells. We interpret these data that possible contamination with mouse DNA is ubiquitous.
Mm-hmm.
Blah, blah, blah. They don't really pin it down, but they say it can happen anywhere. I'm reminded of when Vincent and I visited Eric Delwart in San Francisco. We were
Mhm.
riding around the car with him talking about deep sequencing. And as I recall, his comment was, DNA is really a problem. It's everywhere.
Mhm.
So it's not hard to get stuff contaminated with DNA.
Mhm.
I think one of the Coffin papers in the discussion suggested that they had traced it to either calf serum or phosphate buffered saline.
Mhm.
And was it that paper or one of the others? Somebody said they had also looked into the possibility that it might be the heparin in the vacutainers that were used for collecting the blood.
Why would that be?
I'm not sure, but
Yeah, you're right. There is a collected in heparin tubes, rendering the anticoagulant also likely a suspect.
What's the source of the heparin? Do we know?
They don't say. They point out. You're right, Alan. They point out in fingering the fetal bovine serum. They say, after all, it's got bacteriophage DNA in it.
Yeah.
That contaminates the serum during the slaughter of the cattle. So.
John also feels that the places that make, phosphate buffers and so forth, they have mice running around at night and
That's right.
they contaminate. Someone posted on the TWiV Facebook page. I heard Drs. Rein and Coffin talk at a symposium in November about XMRV contamination. Dr. Coffin went as far to say that his lab found XMRV sequences in unnamed supermarket cereal brands.
Yes, it's a serially transmissible agent.
Good.
I think if you have the name Alan,
that's what it is.
You're a good jokester. That's very good. Is that true, Alan Rein, that he said in cereal?
Yes. John told me that also, that some cereal tested positive. Yes. Mm-hmm.
So this is a problem.
The fourth paper makes a phylogenetic argument. Is that correct? For for questioning the link between
origin.
XMRV. Yeah, the origin of XMRV.
So they worked with this 22RV1 cell line, which you had mentioned you used in your study.
Sure.
So, and you said it was infected with XMRV. So when was it infected? Do we have any idea?
Well, we don't know for sure, but this is a cell line that was made by passing prostate tumors in nude mice, as like many human cell lines, tumor cell lines. It's a reasonable possibility that it got infected with a murine leukemia virus when it was passed in mice.
Do you have any idea roughly when this was done, this implantation into nude mice?
A couple of decades ago, I think.
So the XMRV has been around a long time.
Probably.
Yeah. So what they did in that paper was to sequence XMRV from this cell line and compare it to the diversity of all the human sequences. And they said, well, it's more diverse in the cell line than in people. Therefore, this can't be circulating in people.
Mm-hmm.
So what do you think about that?
I'm not an expert on this, but it seems to me like I'm not sure you have to draw that conclusion from those data.
Mm-hmm.
I would probably be more conservative about drawing inferences from it.
Yeah. Yeah, I initially was convinced by that, and then after thinking about it more, I realized that it wasn't a smoking gun.
Mm-hmm.
Yeah, I put up a blog post about this, which Towers, the senior author on this, is quoted making very definitive statements about it in a press release, which was the source of many of the news articles about this. But in the paper, while being peer-reviewed, of course, the statement is qualified much more carefully. That this is consistent with all of them having been laboratory contamination, but it doesn't absolutely nail the case.
Right.
So what we're left with these four papers is that they point out ways that contamination can be an issue, but they do not impugn any of the previous studies, right? Necessarily.
Right.
So what it says is that we really need some sort of standardization of approaches, protocols, a package of appropriate controls, etc., and a lot of trading of samples and ideas to sort all this out.
Exchange of reagents, exchange of samples.
And I think these articles highlight the types of protocols that you would need. For example, you would have to, and a lot of these things are going to be straightforward controls that you can include, looking for IAP instead of just mitochondrial DNA. But some of them, I imagine, are going to be logistical nightmares. I mean, the issue with samples taken at different times by the same lab giving different results, that suggests to me that you would have to interweave your cases and controls and maybe even have the blood samples taken by the same technician in the same room with the same lot of vacutainers. It's a lot to organize, right?
Well, yeah. I mean, but a genuine positive would be positive for virus and not for IAP sequences, for example. You can critically scrutinize each sample, really, it seems to me. It's also important to have a sufficient number of negative control assays, not just 50 clinical samples and one HeLa cell DNA control, but a substantial number of HeLa cell DNA controls, because maybe your assays will give you false positives, let's say 10% of the time.
I mean, would it be better to use immunohistochemistry instead of PCR?
I think multiple assays are better than any single assay.
You mentioned that someone, I suppose the NIH is coordinating some kind of study to determine what kind of assay or assays would be used. Do you know what's being considered? I mean, is PCR, IHC, are they all part of it, or there's something else?
It's probably mainly PCR and RT-PCR.
Can you imagine a situation where somebody would be PCR positive and immunohistochemistry negative? Is that a possibility?
I think that's a possibility, that you could have viral sequences integrated into cellular DNA, but not expressed.
Yeah, okay.
That's actually an interesting point, because Silverman's group has identified, and I wanted to ask you about this, Silverman's group has identified proviral human DNA junctions in prostate tumors. And I wonder if you thought that was a reasonable study, and why hasn't it been done for chronic fatigue syndrome samples?
I think that is a very definitive way of showing you really have an infected cell, is if you can show viral sequences linked to human cellular sequences. There's no way mouse DNA contamination will give you that result. So I think that's a very important result, and I would say it needs to be reproduced in other labs. And why hasn't it been seen in chronic fatigue? I have no idea. And I believe that Ila Singh, for one, is working quite hard to demonstrate integration in her prostate samples. And I believe it takes a lot of DNA, and you don't get much DNA from prostate tumor biopsies. So that's a logistical problem.
There were 2 points brought up in various comments made, and one was in the original Science paper from WPI. They found antibodies to XMRV in patients' sera, and some individuals said, doesn't that prove that these individuals are infected with the virus? So how would you address that?
Well, one important answer is it needs to be reproduced in other labs. I guess that's my answer.
And also the fact that they transmitted virus from from blood to cells in culture. I was also told that that's proof that there is virus there. I guess you would have the same answer, right?
Yes, and also, I mean, obviously, if you have XMRV floating around in your lab, you can get false positives in virus culture assays.
Sure. Right.
And what is your assay for having transmitted the virus?
That's straightforward. I mean, the virus, there are permissive cells, such as this cell line LNCap. The virus can replicate in these cells, and you can easily detect the replicated virus by Western analysis or reverse transcriptase assay or
Got it.
PCR, or we also have biological assays with reporter genes that detect replicating murine leukemia virus.
Just out of curiosity, does the virus cause any cytopathology, any morphological changes in the cells, or kill them?
No. Like most murine leukemia viruses, the cells look just the same and grow just the same, as far as I know, as uninfected cells.
Okay.
Another comment was made, if the previous results are due to contamination, why would it be more prevalent in disease samples compared to controls?
Well, I don't know. Is it possible that the disease samples have been handled more than the controls? I don't know. I don't think we know the answer to that, but...
Well, and in the new set of papers, there's also what to me was a very disturbing result that they had, as I said, 2 sets of samples taken at slightly different times. They had their CFS samples, I think it was, and then they went and they took their control samples, and they started looking for XMRV, and they started finding it in one set of samples and not the other. So it may depend when you took the samples and under what circumstances and how much mouse DNA was floating around in your lab at the time.
Right.
Which is why I brought up the issue of interweaving your cases and controls in a clinical study, because then you could potentially get around that issue.
Yeah.
All right. So let's summarize what all these papers mean going forward. I mean, what is next with XMRV, prostate cancer, and chronic fatigue syndrome?
Exchange of samples, testing samples in multiple labs. We need assays that people can agree on.
Mm-hmm.
At present, Lab A does its assay and says, I found it, and Lab B does a different assay on a different sample and says, I can't find it, I don't find it. So who's right? You can't get the answer that way.
But you agree that needs to be done, right? That needs to be.
So that's what's next, is we need to
mm-hmm.
exchange reagents and exchange assays.
So I had said that large case control studies are needed to find the prevalence of this virus in the population, and you don't agree with that, right?
I think that's premature. And I mean, until we have validated assays, so what are you going to do for the large epidemiological study? Whose assay are you going to use? What lab are you going to use to do the assay? It's premature, and I feel it could cost a lot of money, and as I hope is clear, I think it's possible that the whole thing is a mistake, that there is no XMRV in the human population. I think that is a possibility, in which case, if that is right, then the less money we spend on it, the better.
But is it also possible that it is a virus infecting people?
Of course it's possible, yes. But and so we need good assays before we start a large case control study.
I think that's reasonable.
Right. So the assay has to be reproducible in every lab that's using it, and then you can go to the large cohort.
This Baylor study that you picked out that was published, I think, a while ago, like in October or something like that, was accompanied by a commentary that you also pointed out that had, in the commentary, they suggest several things that need to be done. One is standardization of detection assays. The other is prospective epidemiologic surveys. I guess these are in order. Well, not really in order, because I guess you'd say that that's probably premature. You got to get the assays right. Sharing of reagents and samples, comprehensive and rigorous phylogenetic analysis, and development of trackable models. But it seems to me that standardization of the assays and the sharing of reagents and samples is the first step.
Mm-hmm.
And I love the subtitle of this commentary article by Kearney and Maldarelli.
Mm-hmm.
The title is Reach for a Scorecard, Not a Prescription Pad.
Right.
Anything else, Jim?
I would like to make one further comment, and that is about the level of virus in positive samples. So our general routine in our PCR assays was to assay 100 nanograms of DNA. A cell has 6 picograms, and so 100 nanograms is a large number of times 10 to the 4th cells worth. And when they were always negative, we were sometimes told, oh, well, you have to assay more DNA. You need to assay a microgram to see it. In other words, 10 times more sample.
Mm-hmm.
And, well, that's possible, although it's not easy to get a microgram of prostate cancer DNA. But anyway, that's possible. You can always say you didn't assay enough material. The incidence of infected cells, the frequency of infected cells is so low that you have to assay more cells worth in order to see a positive. That is possible. That is always possible. On the other hand, from everything we know about how gamma retroviruses cause cancer, all the cancer cells should be infected. And if you say, well, 1 cell in a million is infected, then I don't think that's compatible with known mechanisms of tumorigenesis by murine leukemia viruses. And so, it's possible that the virus is present at extremely low levels. But in that case, I don't think it has anything to do with the disease.
Mm-hmm.
That would be my strong expectation, is that it's just some kind of passenger if it's at such low levels in the tumor samples.
Right. So that's an interesting point, that once we get to the point where we can reliably make good statements about whether or not there's virus present, then you approach the next step as if there is virus present, is it consistent really with a disease state?
Mm-hmm.
An association isn't sufficient.
Right. To put it another way, you have to distinguish the two questions. Is it present in disease tissue from what is its relationship to the disease? Does it have any causal role in the disease? Those are two
right. Or is it
questions.
just an opportunist that ended up there?
Right.
Alan, would you stay with us while we do a few email questions?
Sure.
Because some of them are right up your alley, I think. All right, the first one is from Trevor, who is a PhD student at the University of Toronto in physics, listening to TWiV since last Christmas, when his brother, who is a graduate student studying mass spec determined protein sequences with massively parallel computer systems, recommended the show to me. I've enjoyed every moment of the show, which has helped deepen my understanding of viruses and topics related to them. I had a comment on Alan Dove's response to the letter about the media and dissemination of scientific results. Now, to remind everyone, this was TWiV 105. Trevor asked, like the Wakefield saga, it seems as though the Whitmore-Pedersen study has given a glimmer of hope to a patient population suffering from a difficult-to-diagnose disease. As we saw with the Wakefield study, even though several studies disproved the association between the MMR vaccine and autism, many people were reluctant to believe it. So he says now, while agreeing with Alan's response, I think he may have missed the exact point of the letter or my interpretation of it by discussing the results of the study in such detail. While the XMRV studies seem much more credible than the Wakefield studies, I think there are similarities in how the studies are being used by advocacy groups and individuals in the public. Some people in the CFS community have pounced on the original result in the same manner that the autism anti-vaccination jumped on the Wakefield study. One only has to do a quick search on YouTube to find videos for XMRV to see this. In these cases, I'd suggest that the link has become an article of faith rather than a reasoned interpretation of the data, and that no negative article will change this. In this way, XMRV is quite similar to the MMRV vaccine autism link. Is there a better way to spread scientific discoveries and encourage critical thinking? Maybe it will take a complete retooling of education systems to help people better understand how science works.
We've had that discussion before.
Yes.
I agree.
How much science do you teach?
I agree.
We need We need better science teaching in the curriculum. And the critical phrase here is critical thinking.
That's right.
Yes.
Teaching people critical thinking.
That's right.
Now, that's much more important than the facts or anything else you can teach people is how to think.
And whether the data agree with your preferences has nothing to do with whether the study was well done.
Right.
Mm-hmm.
As we discussed last time, though, what to teach. I mean, we agree that we need to teach better science, but exactly what to teach them, we don't agree on. We don't really know.
Right.
And we discussed that last time. Anyway, thank you, Trevor. Trevor he seems to be at the University of Toronto and also somewhere in Germany.
Maybe they have a branch campus or something.
The next item up is our artist friend John from Australia, who has sent a drawing of Rich, the last of the 3 caricatures, and we'll post that online.
I showed this to my granddaughter, who is 2.5.
Yeah.
Actually, as a group, we showed this to her and said, "Who's that?" And she said, "Dude," which is me.
That's you.
Okay, so
Dude.
so she recognized it.
Your granddaughter calls you dude.
That's
great.
correct.
So we'll put that up. The next
So
letter.
good job, John.
Next letter is from Shannon, who writes, "Hello, Team TWiV. I'm writing today because I'm in the midst of listening to episode 100 and your discussion of why young people don't go into careers in science. I'm forwarding an email from venture capitalist Larry Bach to a good friend of mine who is very involved in science outreach to young people and the general public. You can read more about the USA Science and Engineering Festival that Larry is organizing in the email below. But the key line that I want to share is society gets what it celebrates. As a culture, we celebrate movie stars, rock stars, and athletes, and we generate a lot of them, but we don't celebrate science and engineering. A great way to start celebrating science and engineering is this 2-day festival being held on the Washington Mall October 23rd. See this link for more details. I think it would be a great event for TWiV to publicize so they can attend the event or forward the information on to people in the D.C. area. And Shannon is a graduate student at the Salk Institute.
That would have been a great event for us to publicize.
I'm very sorry that we are so late in publicizing it, but I thought we would put the link up anyway.
Well, hopefully there will be events in the future.
Yes.
Yeah, I think they're planning to do this as a regular thing.
Does anyone know Larry Bach?
No, I do not.
No.
I've heard the name.
Well, it's a good point. We should celebrate science, right?
Mm-hmm. Yes.
But you have to understand it first. Otherwise, you don't know what to celebrate, right?
Mm-hmm.
The next one is from Mitchell. Hello, I was just catching up on some back episodes and heard your conversation in TWiV 51 regarding vaccination of fish and how there is some poor guy in Chile inoculating 300,000 fish by hand. I recently learned of this company, Microtech, which has a machine for doing just this. The company was recently acquired by Pfizer, so they obviously have something good. There's still a lot of manual labor involved, but the machine does the heavy lifting. So he sent a link, but it seems to be broken.
Yeah, I think it's broken because the original link was before the Pfizer takeover or something
I see.
like that.
So apparently there's some mechanized way of inoculating fish, but maybe if you hear this, Mitchell, you have a new link.
Well, the solution really is to put the needle onto a fish hook and give that to Dick. So as he goes around the world fishing everywhere.
Apparently he's going around so much, yeah. The next one is from Yasmin, who writes, I love virology, and although I'm not a virologist, I constantly read about it, and recently I read an interesting article. Not quite sure I understand the whole mechanism. Question. I read an article about using cyclotriazodisulfonamides, CATA compounds, to downregulate CD4 receptors up to 90%, and therefore block HIV adhesion to CD4 positive cells. It's been shown to be very effective in decreasing HIV infectivity and also works synergistically with other HIV meds. Here's what I don't understand. So one would have to take this med continuously to prevent new virions from attaching, and the drug will never get rid of already infected cells. If this is the case, would it be a practical treatment for patients to take for the rest of their lives? Plus, I'm sure CD4 receptors are there because they serve a role in binding substances. Blocking them would not affect health? Alan, probably this is up your alley, right?
Yeah. I don't know blocking 90% of CD4 receptors would do, but in fact, all of our anti-HIV therapeutics work just as you described. They have to be present in the body continuously to block ongoing replication of the virus. That's the only way they suppress the virus in the patient. And so you have to take your antiretroviral medications on schedule every day without fail for the rest of your life. Yeah.
This compound is, well, CD4 has its own function in cells, so yes, it could be that anything that blocks it would have side effects. But if I'm not mistaken, there have been attempts to block CD4 as a therapeutic approach, and none of them have really worked out.
Well, yes, and CCR5, the co-receptor for transmission of the virus. There's been a lot of work on drugs that block that as a way of blocking viral entry.
Yeah, the CCR5 drugs are actually on the market, right?
Yes.
So that does work, because as we know, and some people don't even have CCR5, and they're
That's
fine.
right.
So that is a target. But CD4 is important in immune responses, so I can't imagine that you would be able to block it.
And a receptor blocking strategy like this, as she points out, it works synergistically with other anti-HIV meds, but I wouldn't expect it to work on its own.
Well, yes. I mean, none of the drugs work on their own because the virus always finds a way around them, always mutates to become resistant. And so the only way we can suppress virus replication is with multiple drugs simultaneously to keep the replication so low that it doesn't have a chance to mutate.
I would like to point out that Yasmin is right on top of this.
Yes.
Very, very good questions. She's right on the ball.
Even though she's not a virologist, right? Must have had a good science education, Yasmin.
That's right.
The next one's from Rick. I enjoyed listening to episode 103 with Dr. Tan. I appreciated his explanation of flu mist. Pretty cool. So if I understand correctly, the attenuated virus in flu mist is engineered to produce HA from the H1N1 pandemic strain, HA from H3N2, and HA from type B. Yes, separately. 3 different strains, right. The HA and the NA, I believe, are put onto this flu mist backbone. Also, a second question, kind of off topic. Why do adeno-associated viruses, when used as a gene delivery vector, require a helper virus, and what is the role of the helper virus? Dr. Condit, is that up your alley?
Well, I can answer it. I would say that they don't require a helper virus in the act of gene delivery. Okay, they require a helper virus either in their normal growth or in making the vectors. And that's just talking about the normal virus. It needs the cell to be turned on and replicating DNA in order to replicate its own DNA because it needs the cellular replication, DNA replication machinery. And the role of the helper virus is to do just that, to turn on the cells to replicate DNA or to make DNA replication enzymes. You need that to grow the virus or the virus vector. But once you do that, you purify the vector, and in fact, it can't replicate on its own. It's lost. It's been deleted of most of the guts of the virus, and you deliver that as a vector just to deliver the gene.
I appreciate you and your colleagues taking the time to put this awesome podcast together. TWiV and TWIP, the Parasite podcast, not photography, help my commute go by quickly. Well, one more question, perhaps for Dr. Condit. Many TWiVs ago, Dr. Condit described the antiviral mechanism for acyclovir and gave a nice discussion on viral TK. I've read that acyclovir can irreversibly inhibit viral DNA polymerase. In addition, competitive inhibition and chain termination. Any insight as to how this drug can irreversibly inhibit DNA polymerase?
So Rick sent along with that a paper, a JBC paper from 1984, titled "Acyclovir triphosphate is a suicide inactivator of herpes simplex virus DNA polymerase." And I punted on this, and I sent it to my buddy Dave Bloom, who's a herpes virologist who has been on TWiV before. And it turns out, luckily, that Dave's wife is an enzymologist who works on DNA repair enzymes. So together, they kind of went over this, and I'm glad I sent it to them. basically Dave's wife was not convinced that paper totally nailed down the notion that acyclovir was an irreversible inhibitor of the polymerase. She wanted an additional experiment in the paper where they would isolate what they felt was a complex of acyclovir and the enzyme that was in an irreversible inhibition configuration, and then challenge that with a fresh template. And she didn't see that experiment done correctly there. So she didn't know how they could make the distinction between that and a chain termination mechanism. It was apparently only 5 years later or something like that the chain termination mechanism was really pinned down. And by the way, for those who've forgotten, this is a compound that it's an analog of a normal base, but it's missing part of the sugar, so that if it's incorporated into DNA, it doesn't have what's necessary to incorporate the next base. So DNA replication stops at that point, and that's how it works. And apparently the dogma in the field now is that indeed that chain termination mechanism is the major mechanism of inhibition. So that's what I found out about that.
Good. Next email's from Julia. Hi, I've been listening to the podcast on my dog walking sessions. Quick question about a throwaway comment from, I think, the CFS podcast. There was a mention of CCR5 gene therapy for bone marrow transplantation in HIV. I cannot find anything on this being a reality, just a theoretical concept, but the comment came across as if it was actually being done. Can you clarify at all? And Julia seems to be a physician in UK, a consultant in pediatric immunology and infectious diseases. Yes, in fact, we talked about this on TWiV 88. It's based on a science paper called RNA-based gene therapy for HIV with lentiviral vector-modified CD34 positive cells in patients undergoing transplantation for AIDS-related lymphoma. They basically take peripheral blood hematopoietic progenitor cells and they put in inhibitors of HIV and the CCR5. Then they put those back in the people. And I think they did it in 3 patients in this paper. I don't know if there have been other reports of this since then. You know anything about that, Alan?
I think that people have also taken cells from people and put in the CCR5 deleted gene.
That's right, yes.
Which is a dominant negative inhibitor, and then put those back into the patient, and those cells are resistant to infection by HIV. I think this seems to work very well, except of course it's incredibly expensive and complex, but I think it's very promising as an approach.
All right, the next one, the last one, is from Neil. Thanks for the great podcast. I'm catching up on recent episodes and was listening to episode 103 with Dr. Tan talking about flu vaccine. I used to work at Medivune while it was still called Aviron, and so could not resist writing to correct an error, which was Dr. Tan saying that FluMist is an engineered vaccine. In fact, it was attenuated the old way by serial passage in vitro at low temperature. My project was actually to engineer a separate vaccine strain, but this was shelved once the FluMist strain, aka cold adapted, was licensed by Aviron from the NIH. Anyways, since some folks get nervous about genetically engineered organisms, I thought it would be important to let listeners know about this error. All right, thank you, Neil.
Except I think the issue here is not just that the original vaccine strain, as I understand it, the original vaccine strain was attenuated the old way, so they have this cold-adapted strain. But then what's genetically engineered is each new season's batch of antigens gets inserted into the virus.
So the question is, how are those put in, by reassortment or by genetic engineering, right?
Yeah, and that's genetic engineering, actually, at this point.
Is it?
Yes.
Yes, they clone them in, is my understanding.
So they use the 8 plasmid recovery system, and they...
Yes.
Yeah?
Yes. They originally, my latest information says originally they had to do it by co-infecting and then sorting through the reassortments.
Right.
Okay. but they've since gotten approval from the FDA to do it by the engineering the 8 plasmid method, so that they can actually deliberately construct the viruses they want.
Right. So at the risk of making people nervous about genetically engineered organisms, in this context, it's a really good thing.
Right.
You know, it's interesting. The inactivated vaccine is still made the old way. They take a high egg-yielding strain and co-infect cells with that and the current seasonal strain that they want to use. And then they go through all the reassortants, as you say, Rich, and look for the ones with the HA and the NA of the right strain. So they don't actually use reverse genetics for that. So I don't know why it would be done for flu mist, if you guys say so.
Well,
yeah, that is, as I say, that is my understanding, and I guess Rich.
That's my understanding. I've got my first graduate student actually works for MedImmune, and he was at Averon when they were developing all this stuff, and is now in quality control, and has been with the flu mist thing from the ground up. And if I'm remembering correctly, our recent correspondence with him inquiring about this very issue, he said, no, they now do it the genetic engineered way. And then he was shocked at how easy it was to get it through the FDA.
Okay. Do you have a link or a reference that we could post for that?
What I can do is I'll ping him again and see if I can find that.
Okay? All right. Let me know what happens.
I will do that.
Let's do a couple of picks of the week here. Rich, what do you have?
This is viral, so probably everybody has seen it by now, but it's just too cool. An iPhone app called WordLens that is a text translation thing, and I got a link to a little YouTube thing that advertises it. So I can't put this on my phone. I'm going to have to upgrade my phone. I've only got a 3G. I need either a 3GS or a 4 to do this, but you can take your camera with this app and point it at a sign that's in English, and the English is translated to Spanish. They've got a couple of languages, but it translates the sign. You got to get actually, the app is free. They sell you the translators, right? So it's kind of like razors. They give you the razor and sell you the razor blades, right?
Or printers.
Or printers, yeah. They sell you the ink for a fortune, right? So, but it's very cool. And the demonstration on this YouTube link is really cool. You can do English to Spanish or Spanish to English, and I think they got another language as well. First time I thought it was some sort of joke, but apparently it's for real.
So you're in a foreign country, cool. You want to see what a sign says, you can do this.
You can do that, or you can read a menu.
I see. Okay.
I read some of the reviews on it, and apparently they even say, it's not perfect yet, but it's a step in the right direction. And apparently some of the people writing reviews were unsatisfied with it. Others say, get a life. This is amazing. Come on, lighten up.
That can be taken two ways, of course.
Yeah. I think it would be really useful in Japan because I couldn't read any of the signs there.
Absolutely.
At least
they need a Japanese one.
At least in Europe, you sort of get an idea, but
yeah, well, with the characters so unfamiliar, you can't even You can't even make up a name or something like it. It's completely crazy. All right,
cool. That's great, Alan.
My pick is a little off the wall, but for people who have left the lab, one of the common themes among folks I've talked to who've been lab researchers, and then they go out, they do something else, and they say, I don't really miss the frustration and the grant and paper writing. I could do without all that. But what I really miss is actually sitting down and doing an experiment. You mix some chemicals and you get a reaction and you get some cool result. And so one of the ways I recapture that is I still photograph with film occasionally. And I have just discovered that you can get these little kits of chemicals to process color film at home. And for $20, you get this little box of stuff, and you mix up the powders, and you develop. You need some basic equipment, but it's not. You don't need a full darkroom or anything. And it's a very, sciencey type of activity. It's a lab type of activity. You go through this process, and you can't see. Of course, you've got it in this tank. You can't see anything, and then the very end of the process, you pull out your film and you have color photos. and it's just a really cool experience. I can't.
Very cool.
Yeah, I had done it with black and white before, but for some reason, doing this with color, it just really it hits those buttons, and it's.
I recall color film developing as being very complex, very touchy.
Yes, that was my memory of it too. And when I discovered this thing and started reading reviews of it, I figured I'd give it a try because for $20, why not?
Right.
And it turns out the only thing that's really even somewhat touchy is the developer temperature, the first step. And that you only do for 3 minutes. So all you have to do is have a good thermometer and get the temperature right for 3 minutes. And you can generally do this in the kitchen. And then the rest of it just goes straight through. And apparently this kit was originally developed for reporters, back when reporters shot on film, photojournalists, and they could use this in places where they didn't have a print lab. But now it's used by people like me who are just eccentric hobbyists in their homes.
So then you take the film and you scan it into your computer, right?
Yeah, exactly. If you do exactly, yes. If you have a dark room, you could actually do the whole printing process, but that gets much more complicated.
So you must have these rolls that you thread the film on, right?
Yeah. So you have to have a developing reel and a tank and a what's called a changing bag, which is it's like a black shirt with the neck sewn shut and the zipper on the bottom. And you put everything in the changing bag, and you could stick your arms in, kind of like a glove box.
Right.
So you can load everything in total darkness. And
well, there you go. Anybody does it, let us know.
Yeah. Well,
my pick comes from John, our artist in Australia, who made all these nice caricatures of us. He has a pick of the week, POTW. Now we have a great acronym for pick of the week. This is a blog by a fellow named Derek Lowe. It's called In the Pipeline, and he is a chemist who has worked for several pharmaceutical companies on drug discovery, including schizophrenia, Alzheimer's, diabetes, osteoporosis. He has just a terrific blog, really thoughtful, and I've been reading through. He has an interesting article here summarizing a study in PLOS ONE on the problem of widely varying reviews from different referees for the same exact paper, which we all are very familiar with. So a very thoughtful blogging, science blogging. He's a chemist. He seems to be a really good chemist. So check that out. But also he has another link that John sent us. It's called Things I Won't Work With. This is also by Derek Lowe. And this is chemicals he won't work with for various reasons. And this is really funny. So nitrotetrazole oxides is one of them. So anything with 4 nitrogens I stay away from. So it's pretty funny.
He's got a terrific sense of humor. This is chemistry humor, you know?
Yes.
It's
so
incredible.
This is someone who clearly knows his chemistry. I could never write any of this, obviously. I don't think I could do it with viruses even. So he knows his stuff, and he's really funny. So thanks for that, John. And do check that out.
What this reminded me of that's really entertaining is, we're concerned with laboratory safety all the time, but, we concern ourselves with getting infected with a pathogen or handling some chemical that's going to give you cancer or something like that. This guy's a chemist. What they worry about is stuff that's going to blow up.
Yeah. Yes.
And so they must sit around telling stories about stuff that blew up all the time.
Yeah, and I think
Great stuff.
that's what's here, a lot of that sort of thing. Why I don't work with this, because it's going to do this or that.
As a matter of fact, he's got a story later on, Max Gergel's memoirs, about stuff that blows up.
Yeah, so
Good.
John says his Things I Won't Work With is hilariously funny while being, sort of educational. So thanks for that, John. So thanks for your drawings and for that pick of the week. And that'll do it for another TWiV. Don't forget, you can find us on Facebook, facebook.com/thisweekinvirology. You can hear us on iTunes at the Zune Marketplace. If you do subscribe, leave a comment. That helps keep us on the front page of the medicine directory. You can also listen on your iDevice or your Android device. Microbe World makes an app for those devices. You can find it at microbeworld.org, or you can go over to twiv.tv, play the episodes, download them, and read the show notes. And as always, send us your questions and comments to twiv@twiv.tv. Alan Rein is at the NCI. Alan, thanks for joining us today.
Thank you. It's been a pleasure.
I appreciate it, and I know it's hard to keep a phone on your ear for over an hour, so thanks very much.
That's all right.
Alan Dove, thank you.
Always a pleasure.
AlanDove.com. Have a good trip down south.
Yep.
Rich Condit is at the University of Florida, home of the Gators.
Great to be here. Have always a good time.
You're not going anywhere, right?
No, some of the people are here. Sarah and Joey and Harper are here.
Very good.
So it's all good. It's great.
Well, have a good holiday down there in Florida. I'm Vincent Racaniello. I'm at virology.ws. Everybody, happy holidays. We are going to record one more TWiV this year on December 31st, where we'll talk about 10 virology stories of 2010. And that will go up on the 2nd of January. So, happy holidays, happy new year to everyone. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.